The idea of human cloning has long captivated our imaginations. From science fiction novels to blockbuster movies, the concept of creating an identical copy of a human being has been explored and debated for decades. But what was once confined to the realm of fantasy is now edging closer to the realm of possibility. The birth of Dolly the sheep in 1996, the first mammal cloned from an adult cell, shattered scientific barriers and ignited a global conversation about the ethics and implications of human cloning. Dolly's arrival proved that cloning was no longer a scientific impossibility. It thrust the topic into the public consciousness, raising profound questions about the very nature of life and our ability to manipulate it. Could we or should we apply this technology to humans? The world watched with bated breath, waiting for the other shoe to drop. Rumors of secret cloning attempts surfaced and faded, fueling speculation and anxiety. Yet over 2 decades later, no confirmed case of a cloned human has ever materialized. This begs the question, if human cloning is scientifically feasible, why haven't we seen it yet? The answer, as we shall explore, lies in a complex web of ethical concerns, societal anxieties, legal barriers, and a shifting scientific landscape. To understand the complexities surrounding human cloning, we must first delve into the science behind it. The most widely known technique is somatic cell nuclear transfer, or SCNT, the very method used to create Dolly. SCNT involves replacing the nucleus of an egg cell with the nucleus of a somatic cell, which is any cell other than a sperm or egg cell. The egg, now containing the genetic material of the donor, is stimulated to divide and develop into an embryo. If this embryo is implanted into a surrogate mother, it has the potential to develop into a cloned individual. While SCNT has been successful in cloning various animal species, it remains a highly inefficient and unpredictable process. Cloned embryos often suffer from developmental abnormalities, leading to miscarriages, stillbirths, and severe health problems in cloned animals that do survive. Another avenue for potential human cloning is through induced pluripotent stem cells, or iPSCs. Discovered in 2006, iPSCs are adult cells that have been reprogrammed to an embryonic-like state, capable of differentiating into various cell types. While iPSCs hold immense promise for regenerative medicine and disease modeling, their potential for human cloning remains largely unexplored. The prospect of human cloning raises a myriad of ethical concerns that strike at the core of our values and beliefs. One of the most pressing concerns is the potential for harm to the clone. Given the high failure rates and health risks associated with animal cloning, attempting to clone a human being would be a highly irresponsible and ethically questionable endeavor. Beyond the physical risks, there are profound psychological and social implications to consider. What would it mean for a cloned individual to live in the shadow of their genetic original? How would their sense of identity and individuality be affected? These are uncharted waters, and the potential for psychological harm is immense. Moreover, the possibility of human cloning raises concerns about eugenics, the practice of selective breeding to improve the human species. Cloning could be used to select for certain desirable traits, such as intelligence or physical prowess, leading to a new form of genetic discrimination and exacerbating existing social inequalities. The idea of human cloning has sparked widespread public debate and controversy. Public opinion polls consistently show that a majority of people oppose human cloning, citing ethical and moral concerns. The image of scientists playing God and creating life in a lab evokes feelings of unease and distrust in many. The media often portrays human cloning in a negative light, fueling fears about a dystopian future where clones are exploited or treated as second-class citizens. These fears, while often rooted in science fiction, tap into deep-seated anxieties about the rapid pace of technological advancement and its potential impact on humanity. The lack of a social consensus on human cloning creates a significant barrier to its advancement. Scientists and policymakers are understandably hesitant to pursue research that is widely viewed as unethical and potentially harmful. The ethical and societal concerns surrounding human cloning have led to widespread legal restrictions and bans. Many countries, including those at the forefront of biomedical research, have enacted laws explicitly prohibiting human reproductive cloning. The United Nations has called for a global ban on human cloning, citing the need to protect human dignity and prevent the exploitation of women and children. While the UN's declaration is not legally binding, It reflects the international community's strong stance against human reproductive cloning. These legal restrictions, coupled with the ethical and societal concerns, have created a formidable barrier to human cloning research. Scientists who violate these laws risk facing severe legal penalties, professional censure, and public condemnation. Section 6. A shift in focus. The rise of alternative technologies. While human cloning remains a controversial and largely unexplored frontier, the field of biomedical research has made significant strides in other areas, such as stem cell research and gene editing. These advancements offer alternative avenues for addressing many of the medical and scientific questions that cloning was once seen as a potential solution for. Induced pluripotent stem cells, or iPSCs, for example, hold immense promise for regenerative medicine. Scientists are exploring the use of iPSCs to grow new tissues and organs for transplantation, potentially alleviating the shortage of organ donors. iPSCs can also be used to study diseases in a dish, allowing researchers to test new drugs and therapies without the need for human subjects. Gene editing technologies, such as CRISPR-Cas9, have revolutionized our ability to manipulate DNA with unprecedented precision. CRISPR has the potential to correct genetic defects, treat inherited diseases, and even enhance certain human traits. While gene editing also raises ethical concerns, its applications in medicine and agriculture are vast and transformative. Section 7. The future of cloning. A question of why, not how. The question of whether human cloning will ever become a reality is not simply a matter of scientific capability. While the technical hurdles may eventually be overcome, the ethical, societal, and legal barriers remain formidable. The future of human cloning hinges on a fundamental question: why? What are the compelling reasons for cloning a human being? Unless and until we can articulate a clear and ethically justifiable purpose for human cloning, it is likely to remain confined to the realm of science fiction. As we continue to grapple with the implications of powerful new technologies, it is imperative that we proceed with caution and engage in thoughtful dialogue about the kind of future we want to create. Section 8. The unforeseen consequences. A Pandora's box? The potential consequences of human cloning extend far beyond the individual clone. The very act of creating a genetically identical copy of a human being could have profound and unpredictable ripple effects on our understanding of family, identity, and the nature of human existence. Some argue that human cloning could lead to a devaluation of human life, as clones might be seen as commodities or replaceable copies. Others fear that it could exacerbate existing social inequalities, as the wealthy and powerful might use cloning to perpetuate their privilege. The ethical and societal implications of human cloning are complex and multifaceted. It is essential that we engage in open and honest conversations about these issues before we venture further down this path. Section 9. Conclusion. A balance of progress and prudence. Human cloning stands as a testament to the boundless potential of science and the profound ethical dilemmas it can pose. While the technical feasibility of human cloning may be within reach, the ethical, societal, and legal barriers remain formidable. As we stand at this crossroads of scientific progress and moral responsibility, it is imperative that we proceed with caution and wisdom. The decision of whether or not to clone a human being is not one to be taken lightly. It demands careful consideration of the potential benefits and risks, not just for the individual clone, but for humanity as a whole. The future of human cloning remains uncertain. Whether it will forever remain in the realm of science fiction, or eventually become a reality, is a question that only time will tell. But one thing is certain: the ethical and societal debates sparked by this groundbreaking technology will continue to shape our values and beliefs for generations to come.